Thank you for coming out tonight. As Public Relations Head of Neuro and Director of the Committee on Undergraduate Enrichment, it's my pleasure to welcome you tonight for our second Neuroscience lecture series. It's my great honor to be welcoming Dr. Nicolelis to our university and to our club tonight. And thank you so much for coming in such big numbers. I would like to address our thanks on behalf of the Neuro Board to Dr. Nicolelis for coming out from North Carolina to talk to us tonight. And I would like to thank my wonderful exec board, Ben, Christian, Anya, Levine, and Brandon, my really dedicated Q members and other Neuro members that have made this project possible by contributing to many different steps throughout the process. I would just like to inform you that the talk will be followed by question and answers, and then you guys are all cordially invited to take part in a catered dinner outside in the lobby, where you'll be able to ask any additional questions, get to know the club if you're not very familiar with us, and meet other students that share your interests.
I have to apologize. Dr. Nicolelis' accomplishments are so numerous I couldn't have memorized the entire list, so I'm going to read off this paper. Miguel Nicolelis, MD, Ph.D., is the Duke School of Medicine Professor of Neuroscience at Duke University, Professor of Neurobiology, Biomedical Engineering and Psychology and Neuroscience, and founder of Duke Center for Neuroengineering. He's also founder and scientific director of the Edmond and Lily Safra International Institute for Neuroscience of Natal. While Dr. Nicolelis is best known for his achievements in developing brain-machine interfaces and neuroprosthetics in human patients and non-human primates, he's also developed an integrative approach to studying neurological and psychiatric disorders, including Parkinson's disease, epilepsy, schizophrenia, and attention deficit disorder. Dr. Nicolelis believes that this approach will allow the integration of molecular, cellular systems, and behavioral data in the same animal, producing a more complete understanding of the nature of the neurophysiological alterations associated with these disorders. Dr. Nicolelis is a member of the French and Brazilian Academies of Science and has edited numerous books and special journal publications and holds 3 U.S. patents. Thank you all again for coming out tonight, and please join me in welcoming Dr. Nicolelis.
Well, first of all, I'd like to thank the students of the University of Chicago for this invitation. It's actually a pretty special day for me, not only because I'm here, but when I got the invitation, I look at the calendar, and I told over lunch to the students that had lunch with me today that, you know, this is a very special day for my own life, personal and professional life. I arrived in the United States 24 years ago on February 20th. And with me came my first graduate student in Brazil from University of São Paulo, Albert Yu, who helped me to create my first lab in University of São Paulo. And believe it or not, he stayed with me in Philadelphia for 6 months, but then he came and was accepted at the University of Chicago for a Ph.D. and then for graduate school, and also as a resident in the medical school. And Albert was the first neurophysiologist to ever record more than one neuron in a behaving bird in Dan Magulash's laboratory here at the University of Chicago. He was one of the greatest kids I ever met in my life. Unfortunately, he passed away 12 years ago on this day, curiously, the same day we arrived together in the US. So I'd like to dedicate this lecture to Albert's memory because he was really one of the greatest chaps I ever met. So with that note, I'd like to tell you what I came to this country to do 24 years ago. And basically, as I used to tell my students, if I would give this lecture 20 years ago, I probably would be run out of the auditorium very quickly. Because instead of focusing on looking at an individual neuron at the time, that was the norm of what I learned when I was in graduate school. My advisor was someone that always told me that the brain doesn't pay attention to a single neuron. It doesn't mean anything. You know, the brain relies on circuits, populations, to actually do everything that it does. The business of the brain to generate behaviors and models of the external world rely on populations, but we could, barely touch on that subject when I was in graduate school. And my dream in graduate school, before I came to the United States, was to actually achieve things like this, to record the activity of hundreds of neurons simultaneously while animals do something meaningful, meaningful for them, not for us, and on an ethological meaning, I want to say. And I like to start all these talks with this image because this was the first time we recorded the extracellular signals of about 100 cortical neurons simultaneously from 5 different cortical fields in a behaving monkey that was learning to do a particular visual motor task. And we are sampling these signals simultaneously and trying to understand what is that this animal is going to tell us behaviorally before it does, about 400 milliseconds before the animal actually moved the hand in response to a visual cue. And the reason I'm saying that this 10 years ago or 20 years ago was something that would get you out of an auditorium is because, believe it or not, people didn't see the value of doing it. Because for 6 decades, the focus of neuroscience, or at least neurophysiology, was the single cell. And interesting enough, of course, we're not the only ones to believe in this kind of research program. It was this guy that most of you have heard about, the Canadian psychologist Donald Hebb, who wrote this masterpiece, The Organizational Behavior, that I like to say is the most quoted and least read books in neuroscience, because everybody quotes this book to describe the Hebbian learning rule that occupies about 2 paragraphs of the book. The rest of the book is a manifesto in which Hebb suggests that the real true functional unit of the central nervous system is a population of nerves. It is what he called an ensemble, or a cell assembly, that is formed in a given moment in time by the coherent firing of neurons distributed across multiple areas of the brain that come together to underline a particular behavior. And Hebb has never recorded a cell until he went to work with another interesting guy, Penfield, an American surgeon that worked at McGill, and Hebb was his assistant while Penfield was recording human brains and doing neurosurgical procedures. But Hebb had no clue about any of the electrophysiology of populations, because very few people could do anything at that time besides EEG and some field potentials. In fact, at that time, I talked to one of the master men of that period, Werner Mundkessel, who told me that people didn't even consider Hebb because everybody was rushing to record from a single cell. In fact, forgive me, but this is the quote from Dr. Mundkessel: whoever was a real man was recording a single nerve in the cortex at that time. He told me that personally. So Hebb proposed this, and if you read that book, you can extract a theoretical agenda that is still with us today, that is still determining, in my opinion, the agenda for neurophysiologists like myself. They're now capable of recording from lots of cells simultaneously. And some of the very simple questions that Hebb raised, because he was really anxious about it, he had never been able to do an experiment like that, is if I'm right, I mean Hebb, if it is a population that underlies behavior, what is the minimum number of neurons that have to come together in a given moment in time to underline, let's say, moving my arm, or in the case of Albert's work, singing, right, a bird song, or walking. So what is the minimum number? Is always the same neurons? Or it can be any number of neurons recruited from a pool of cells that can underline their behavior. Does that ensemble always does one thing? Or it can do many things at once? So these were all things that you can extract from that book that we're still trying to do. And that's what I'm going to tell you what we have been doing lately to try to answer these questions. I'm going to basically skip a lot of what we did in the early years to show you a paradigm that is, we named in our first paper brain-machine interface. And at that time, when we sent the first grant to NIH, that's a good lesson for any of you that will be inclined to become part of this beautiful thing called science and become a scientist. When we first sent this paper, this data to NIH as a grant proposal, we got back a reply that I published 30 years later saying, Dr. Nicolelis and Dr. Chapin, my postdoctoral advisor, require professional help, at the psychiatric institution, somewhere at Duke University, because people thought that we had lost our marbles. But what John and I were trying to do was to test some of the assumptions of Hebb by creating a paradigm in which, using what you're going to see in a moment, we would be recording large populations of neurons distributed across multiple cortical areas, ask these animals to imagine movements that they like to do on a voluntary basis, and see if we could use a population coding strategies to extract motor commands generated by these ensembles of neurons and make robotic devices execute under voluntary control of these beasts the behavior they were supposed to do to win a reward in a task. And we postulated that if we were right, and Head was right, we would be able to do that with a population of neurons, but not with single individual cells, and that eventually our animals would learn to control these devices just by thinking, relax their bodies, and actually realize that, as a good Brazilian would love to have every day, there is a free lunch. If you can imagine a movement, and the device can realize the movement for you, and you know that that is working somewhere in the environment. Okay? So the entire structure of this brain-machine interface, as we first proposed, is described here. You have a brain, you have implantable devices that allow you to record extracellular currents of individual neurons. You have a real-time platform that extracts motor information, transforms that motor information into digital commands that are transmitted to a device, and that device can be electronic, mechanic, and even virtual, as you're going to see in a moment. That device is in charge of enacting the motor behavior that this brain is imagining, and is providing feedback to this brain so that the brain is aware of how well this device is fulfilling its mission. Okay? All in real time, all within the window of reaction time of the animal that you study. And that is in the order of a few hundred milliseconds, 300 to 500 milliseconds. And as you're going to see, that window is very important, because in monkeys, for instance, if that window is violated, if your computational interface cannot cope with that 1/3 of a second or half a second, the animals will not perform the task. They lose interest in it. Okay? So you really have to be under that constraint. So I also am going to skip the 4 generations of microelectrode designs that we did at Duke since I got there 20 years ago to actually do these experiments. I'm just going to show you the last generations, the last 2 generations very quickly, because I'm not going to talk shop with you guys. I just want to show you the results. But we have evolved from planar systems of microwires that allow us to go to the limit, getting close to 500 neurons simultaneously recorded, and to what we call now a cube, a microelectrode cube, in which we have flexible wires, 10 by 10 by 10. Each one of our shafts contains 10 smaller microwires that can be moved individually so that we can deploy a 3D recording cube. And each one of them has 1,000 contacts, so we can implant close to 4 to 8 in a rhesus monkey brain, and if you do a human, you can implant 10 to 20. So that explains to you how we are basically going up in numbers, and as I'm going to show you, just crossed the 1,000 neuronal recording and got basically close to 2,000 neurons recording an adult rhesus monkey now. And that changes the game dramatically. So this is what happens when you're doing a rhesus monkey. I'm about to tell you that we are actually doing this in a wireless way. So we have microchips that can be implanted on top of the connectors of these cubes, and we can broadcast these signals without cables. So we have entire apparatus that can be implanted and can be left in the head of the monkey for years, 6, 7 years. And in there you have the microchips and the wireless interface that allows us to basically identify individual neurons and thousands of them simultaneously and broadcast about 20 feet or so. So we can do all sorts of interesting manipulations of this neuronal data. Imagine this as 2,000 cell phones broadcasting neuronal activity simultaneously to a device that can gather these signals and do some manipulations with it. So this is what you see in one of the monitors, that you're recording. This is the raw data. These are action potentials, extracellular recorded action potentials. One of the interesting things that we learn over time, if you leave microwires in the brain of animals for long periods of time, years, months, you start getting more than one action potential isolated per microwire. And that's how you actually build your samples. Because about 70 to 80% of those will work. You lose a few that don't work over time. But as you can record more than one per microwire, at the end, the total number of neurons that you get is about 1 neuron per microwire implanted in the brain. Okay? So if you implanted 2,000, you get close to 2,000 neurons overall. And one thing we discovered, this is the latest recording we did just 3 months ago. This is the first time 2,000 neurons were recorded simultaneously. This is time. These are the individual neurons. This is the waveforms recorded from each one of them. This is the top of the head of the rhesus monkey. And that's how it looks like. So you have 56 connectors here that can be plugged in these wireless chips and can broadcast this. So this monkey can be completely free, roaming in a room like this, and basically controlling everything in this room just by thinking. He has appliances that he can control, monkey appliances, if I can talk about that stuff. One thing that is very important about these flexible microwires implanted in the brain, different from other rigid microelectrodes that have been proposed, is that they can record forever. Okay, these are 6 monkeys, and as you can see, this guy is still with us. I like to joke in our lab that we keep monkeys much longer than our graduate students, because we had monkeys that were implanted when Brazil won the last World Cup in 2002. And we're still recording from those guys, and since we're going to play in 2014, I'm hoping to keep them, so we win again. But that's a very important property that is very difficult to achieve with low impedance electrodes like these ones. The reason we get this, in my opinion, is because these wires are flexible. They can move with the brain, and with the deposits of protein and cell stuff in the tips of exposed to these things, you go from a low impedance to a high impedance electrode, and it stays there. So the inflammatory reaction works in our favor instead of against us. And that's what made these things so powerful. And I'm not going to tell you this, but we have done 48 intraoperative recordings in patients with this a modified electrode for human recordings over the last 10 years. And we see the same thing. We can really get very high quality recordings in humans with these things. So how is that the brain-machine interface computational strategy works? So we basically try, poor computational people we are, to reproduce a poor man's spinal cord in these computational programs. So what we do is, we don't fish for right neurons. I don't believe in grandmother neurons, cardinal neurons, mirror neurons. These are all anthropocentric labels that we like to give to neurons, but the brain could care less about them. The brain is about producing behavior. So I actually think that the brain, as you're going to see, is like a little voting machine. It's like a little democracy where populations vote to generate an outcome. And I'm going to clarify what I mean by that. But this is just to show you that whichever sample of neurons we get from these implants, we feed a series of models that can be linear or non-linear, it doesn't matter. But these models are working pretty much like the general idea of how the brain works. They're basically predicting the future. They're using the last 500 milliseconds in time to generate a motor program in the future. So if I'm going to walk, or if I'm going to speak, or if I'm going to move my arms, the brain builds a motor program by counting these volts of these neurons, generate an output that is predicting what the muscles have to do to generate the motor behavior that the brain needs to generate. So that's exactly the strategy of our computational interface. And as I said, there's a rigid time window in which all these computations have to be done, as 300 to 500 milliseconds. If you go beyond that, things degrade. The performance of the interface degrades dramatically. So let me show you a progression of 3 experiments done in the last 10 years. This is one of the first experiments. We actually started with rats, then we moved to all monkeys, and then we did rhesus monkeys and humans very close by, in a difference of a year. But this is the experiment that made a big difference. This is when we got monkeys to play video games. We trained them to use a joystick first to play simple video games, 3 or 4 different classes of video games. And I'm going to show you the simplest one, when they had to move a cursor inside the target. But while the animal is doing that, at that time we were recording 100 neurons simultaneously. And we are playing this through our models and trying to train a robotic arm that was in a different room to reproduce the arm trajectories that are needed for the animal to play the game. Because every time that cursor got inside the target, this animal would have a drop of Brazilian orange juice. And for a Brazilian orange juice, monkeys will do anything for you, even play crazy video games, So they were doing this, and they were receiving visual feedback of how well that arm was learning to perform the task during the training. So you had monkey training, software learning, and machine learning. Hey, you had these 3 stages going on simultaneously. So when we realized that our models, and at that time we were using very simple Wiener filters, multilinear regression, when our coefficients stabilized and they couldn't improve better, and the predictions were at the limit, we stopped the learning of the software. We froze the coefficients. And basically we did a Latin American trick with our little monkeys. Without telling them, we took away the joystick. We turned on the interface, and now to get the same reward, these guys had to figure out that there was no point in moving their bodies. They had to actually just imagine the trajectories, and this robotic arm would now take over the cursor and take it to the center. That was the only way to get the reward. And of course, you could not tell the monkeys that. They had to infer that by trial and error, and that took about 30 days for them to get the gist of what we wanted to do. And so I'm going to show you is the first animal that did that. It became very famous, Aurora. And as you can see here, Aurora is playing a very simple game. She's trying to put that cursor inside the target, and she's a primate, so she's trying to cheat. Before the target appears, she's trying to guess, and sometimes she guessed it right, but she cannot do it just by guessing, because there are 32 different positions in the screen in which this target can appear, so it's impossible to just guess. So she has to master this low inertia joystick and get it to move this correctly inside the target. Well, while she's doing that, we're recording in the good old times of wires coming out of the head and sending these signals to a wall of amplifiers. That it was like a wall, literally. Now it's only boards inside, the head. And the models were trying to get a robotic arm to reproduce these trajectories that you see here so that the animal could play the game. So this is the first day that that transition happened. So this video was the control video, the first few minutes of the day, the night actually. And here, as you can see, the joystick is gone. The interface was turned on, and to our total shock, Aurora kept playing the game. The trajectories you're seeing are the first trajectories generated digitally from signals coming from the brain that allow that brain to play a game without any involvement of the body itself. The body was out of the picture. As you're going to see, we recorded EMGs, muscle activity, from all over the body, and they are all flat at this point. Because I like to say, the brain, a primate brain, got free from the limits of the body. He was now controlling a joystick, was controlling a cursor. In reality, was controlling this big piece of machinery, a 7-degree of freedom robotic arm that was in a different room. And I have to tell you, this is 30 days later, when Aurora actually realized that she had 3 arms. She could play the game using the brain-machine interface to use this hand to move the cursor in there, but she has not lost the ability to use the 2 biological arms that she has. So she could do something else at the same time. She was partitioning her attention and her representation of the body to actually play and do whatever she needed to do. So we start wondering or hypothesizing that perhaps the robot had been assimilated by the brain as an extension of the body representation that the brain carries of that animal. Before I go into that, just to show you that when you make the transition from using the joystick to the brain control mode, that you just saw in the video clip, that's what happens to the EMG activity in the periphery of the body. It goes flat. So monkeys can imagine movements like we do. And they can do that without moving the body. In fact, most of us do that routinely when we sleep. And we go into REM sleep. And we dream about moving, kicking a ball, scoring goals in the final game of the World Cup against Argentina, and becoming a national hero. But most of us do not move when we're doing that. Some of us do. It's a small percentage of people that do, and walk and enact their dreams, scream, do all sorts of stuff. But most of us can imagine movements without enacting them. And we realized that you could unleash the same mechanism in primates under these conditions, under a closed loop control that involves a device. So that was 10 years ago. This is last week. This is a wireless 500-cell monkey recording, living in an enclosure, playing a game without cables, without a chair, without anything. She's playing Aurora's game, but you can see that the precision has improved dramatically, and we can reduce this target 10 times to what I'm showing here. This is the easy task. But she has no cables, no nothing. All the telemetry is in here, and she actually can play this game and play other games that we put around the cage that the plexiglass room that she's living. In fact, we just put together for a competition at Duke of Biomedical Engineering a little cart that Ophelia drives around, our lab, and it can basically go around in a slalom task. And she has a little card on the screen, and she can drive a real wheelchair that is just going around according to what she has to do to get a reward. So that's what 10 years of technology development made a difference. But the results that we obtain with Aurora continue to be replicated, and they are changing, in my opinion, the view that we have of the brain, in accordance to what Hebb would suggested in the past. And I hope if you were here, he would be very happy in seeing this. First of all, as I said, we recorded from multiple cortical areas simultaneously. Areas like the motor cortex, the somatosensory cortex, that's the touch cortex, posterior parietal cortex, SMA, supplementary motor area, premotor area, and according to the old theory that I like to compare to phrenology, people used to think that one function is associated to one area of the brain, and that's it. Cortical, touch cortex, somatosensory cortex only is involving touch, motor cortex only movement. But when we looked at the contribution of individual neurons across the frontal parietal lobe to this task that you just saw, all areas were contributing. The message was widespread. Of course, M1 was the best. Let's say for 50 neurons, M1 was the best area to control or to produce a good prediction of, let's say, hand position. But look at this. The premotor dorsal cortex and the posterior parietal cortex is pretty much as good. They're similar. They have less information per neuron than the primary motor cortex, but the information is there. If you had enough cells, if you extrapolate this curve, if you had 10 times more cells, you would be able to do exactly what we did with M1. So that's the reason we combine cells from all these areas to do what you just saw. More than that, if you start removing individual neurons from these curves, what you see is that below a certain level, things start going bad. By the time you're down to 1, 2, 3 neurons, you cannot do anything. You cannot do anything with a single neuron. And I'm going to repeat for one more time. There is no behavior that can be sustained by a single neuron in the brain. A single neuron in the brain is like a single tree in the rainforest of South America. You cannot explain the ecosystem of that rainforest by looking at one tree at a time. It's impossible. The ecosystem emerges from the interaction of those billions of trees that exist there, not to mention all the little bugs that you find there. I'm from there, believe me. It's not as idyllic as everybody thinks. So a single neuron would not get the brain-machine interface to work, no matter where it comes from, no matter how good the neuron is. It's too noisy. It's impossible to do. So now if you shift from a different parameter, you see that the colors shift. The distribution of the colors shift. It means that different areas are contributing different amounts of information for each of the parameters. So as Hebb said, the same cell assemblies are multitasking. They're able to contribute to different outputs at the same time. So you can see, because these are the same neurons, it's exactly the same pool of cells that we're using. So these are just 3 important things that we saw. First of all, and we call these principles of neuroensemble physiology. We published 10 of those, and I'm not going to give an entire list, but here you see 3 of them that are very important. One is a mass game, neuronal mass game. You need neurons, you need populations to do anything. Second, there is a single neuron insufficiency principle. You don't get anything done with a single neuron. Single neuron does not underlie a percept, it doesn't generate a motor behavior by itself. Third, there's a multitasking principle that I just referred to. Populations can be computing or participating in multiple computations at once. So let's keep going and show you a 4th principle that, again, we knew about it, but we didn't know the extent or the extension of its implication for brain-machine interface or for anything that we do with the cortex. So this is a sample in experiments like you saw Aurora and the new monkey doing. 120 neurons were sampled randomly because, again, I haven't mentioned that yet to you, but that's very important. When we put these wires in there, I'm not sampling any specific cell type. I'm not fishing for a particular cell neuron. I'm getting whatever I get. And it works all the time. It doesn't matter how I do it, how I move it, how I twist it. What matters is how many neurons I get. It's almost like a hologram. If I have enough elements, I see the image. Okay? If I have enough neurons, I can reconstruct the information that I need, but it's not a single cell type whatsoever. Okay? So don't talk about optogenetics with me, the wrong guy. Because I don't believe that you get anything by just selectively going to the cortex and isolating a single cell type and doing that. You're not going to get very far because the brain, again, doesn't know the cell types that we try to impose to it by classifying them as this category or that category. Doesn't make any sense to the brain. But here is a very interesting result, because if you do a random sample of neurons, let's say in M1, and you do a measurement that is called directional tuning, just the preference of neurons for how much they fire in each direction of arm movement. And we know that they like to fire more to one or two directions, and then there is a bell-shaped curve. That's what the z-axis is showing here. This is angle of arm movement, and you see these yellow lines. These are actually compressed z-axis. They're showing the extent of the tuning for direction of arm movement, okay? Don't worry about it. The important thing is there is a diagonal in this graph, which means that every single possible direction of arm movement is represented in a random sample of neurons in the motor cortex. That's what everybody finds. No news about that. But this is the animal using its hand. Now look at this. This is an animal controlling a cursor in a robotic arm like you saw without movements of the hand. So prior to these studies, everybody thought that this graph would be all bluish, no firing whatsoever, because there is no movement of the hand. Instead, the moment you put something in there in the loop, like a robotic arm or a cursor or avatar, whatever, the neurons assume new tuning properties, tunings to the artificial device that is being controlled by the brain. The tuning is somewhat similar to what they had before, but sometimes not at all. But they have tuning. And you can go back and forth between the tuning to the animal's own arm to the tuning of an artificial tool that you put in there. So there is this dynamic plasticity that is going on, as if this tool, as we hypothesized before, was being assimilated as an extension of the body, because neurons acquire tuning to this, external device. So we started looking at this in more detail, and fortunately for us, other people at the same time were getting results that, suggested this is a good line of investigation. For instance, Professor Iriki at the University of Tokyo was recording individual neurons in the parietal cortex in monkeys that basically were being trained to use a tool to basically get food that they couldn't get with their own hands. They couldn't reach and get the grapes. They needed this little rake to go there and get the grapes. And what Iriki showed is that in this area that he was recording, the parietal lobe, neurons had a tactile receptive field in the hand and a visual receptive field in the peripersonal space around the hand. So they have multimodal receptive fields. Well, the moment he gave a tool to these monkeys and the monkeys became proficient, the visual part of the receptive field extended to include the tool. So the neuron now was firing every time something came around the hand or the tool that was holding the hand. That was true in 2D and is also true in 3D. So this was very good, and we basically started looking at this in with more care. So I'm just going to show you a recent study where we moved from, and this is just unpublished, it's just submitted for publication, so we just finished a couple months ago. This is the first bimanual brain-machine interface. And the reason we did it, you'll see a little later, but it's because people said, okay, since we have done unimanual, that's easy. You do unimanual predictions for the left arm, you do unimanual predictions for the right arm, you have a bimanual BMI. And that's not true at all. When the brain engages itself into a bimanual task, the tuning of the neurons that used to control one arm or the other change dramatically. And you cannot predict it by looking at one arm, another, and summing it linearly. It's totally non-linear. Okay? You have to let the brain do the bimanual task to see what is going on there. So that's why we implanted these animals, 2 monkeys, in 2 locations. And now, instead of actually using robotic arms, because it's too expensive and you really don't need it, we created a monkey avatar, a full-body monkey avatar. And we train our monkeys to assume the first person's perspective of that avatar. So this is a whole avatar. Is using the most sophisticated gaming, computing skills that you can get from a high school student. There's programming for you, or undergrad, or because they're the best in the business. So this avatar is totally kinematic realistic, and this monkey plays this game. You know, he has to move the hands and put it inside of the objects that appear floating in this virtual space. You are seeing this as a third person's perspective, so you're observing this from outside. But our monkeys assume the first person's perspective. That's what they see in a computer screen, in a virtual reality room created by them, for them. So they sit in an environment, their hands are occluded, and they see in the monitor these two hands. And they are trained to use their brain activity to control them and to play this game. And the game is, you have the squares. The squares is the initial position. Then they transform themselves into circles or spheres. And the animal has to put the hands inside the spheres in real time, using the brain alone, without moving the body. And that's what they do. They keep tracking the spheres. And they can keep doing that for hours. And that's a real-time brain control bimanual BMI using 500 neurons in each hemisphere. Both hemispheres are being engaged simultaneously. When you look at the curves that I showed to you before, I called those curves in 2002 neuron dropping curves, just because we drop curves statistically just to show that, you know, the same curves were here. You may be looking that they look different because they're like a straight line, but this is a logarithmic scale now. And we found out that this log rule is everywhere. So this is, again, motor cortex, somatosensory cortex, premotor cortex, supplementary motor area, unimanual and bimanual predictions. First of all, you need more neurons to do bimanual. That's a given. Any area that you go, you need more neurons. But you can get very good predictions, you know, in real time with about 500 neurons. What is another major surprise? Because we are talking about 100 million neurons in the primate motor cortex that are there. And I'm randomly picking 500. When we sent that grant proposal to our colleagues that told us that we needed psychiatric help, they are not too far from reality, because we're proposing to record an infinitesimal sample of neurons, and we claim that we could get this to do something meaningful. And it still works. You know, we're talking about 500 out of 100 million. And I have no clue which neuron I'm recording from. You know? So, this is just to show another interesting property. When we started this business 15 years ago, we thought we had to train the algorithms with real movements of the animal. So the animal had to do the task as you saw Aurora doing in a training, and so the coefficients would reach an optimal fitting, and then we'd stop and remove the thing. Well, we realized in this study that that's not needed at all. You can actually train the animals just by passive observation of predetermined trajectories. So you put an animal in a setup, the hands are immobilized, they don't see their own hands, they only see the avatar, and you play a movie with all the trajectories that you want the brain to assimilate. And you play that an hour a day for 3 weeks, and what we saw is that you can get predictions that are pretty decent. Very good. And that gets the animal going without the animal moving a muscle, without any overt movement being produced in the training phase, which suggests that a paralyzed person would be able to take advantage of this, would be able to train his or her brain to actually imagine trajectories that would be useful, as you're going to see in a moment, why I'm talking about that. This is just to show you that this passive observation increases the predictions over training. So by the end of the training, we are getting close to the levels we get when the animals are actually doing the movements themselves, as we did 10 years ago. So what that suggests to me as a hypothesis is that if I would be able to record the cortex of the best soccer player ever to play in this side of the Milky Way, right? Forget about Messi and, you know, Maradona, you know, these guys, but it's not even close. You know, I watched this guy play as a kid in São Paulo, and if you play 1,300 games and you score 1,256 goals, you're darn good, you know? That's 22 years playing 65 games a day, a year. You can play today a day with, you know, Nintendo or whatever, but So this guy was good. So, but there's a famous, you may know if you're a soccer or football fanatic, there's a famous BBC announcer who has called every single World Cup since the 50s, John Monson, who in a famous game in Brazil in '67, England had won the only time they won the World Cup in '66. But Brazil didn't play England, and the coach of the British team was very mad because he didn't feel he was world champion because he had not played Brazil, and I don't blame him. So he organized a whole trip to Brazil to come to Rio de Janeiro in front of 200,000 people. I was 7 years old, I remember this game. Because they needed to play against Pelé in Brazil, so Alf Ramsey was the name of the coach. He would be completely fulfilled in his life as a soccer coach. So they came with the Queen, everybody, and they went to Rio. It was 5-0 Brazil, And John Monson is calling the game. You can find this, and he's calling the game. And the 5th goal, Pelé, who was hurt in '66, he couldn't play the World Cup, gets the ball in the Brazilian penalty kick area and runs the whole field dribbling whatever was in front of him, the referee, the British coach, whatever. And when he gets about 25 meters from the goalkeeper, and this guy, Gordon Banks, is coming out of the goal like a lunatic because, it's Pelé with the ball, Pelé just flipped the ball over the goalkeeper and he scores the goal. And John Monson is saying, this is impossible. The ball is part of his foot. And that's exactly what I predict we would find. Because if you have seen that guy play soccer, or if you have seen a violinist playing violin, or a pianist, or a Formula 1 racer, or in this country you have these crazy games, a golfer with, or a tennis player, those things are not tools anymore after they become proficient users of them. Those things, according to this theory that I'm presenting to you, become part of the body representation. They're extensions of the body. Because through the feedback, visual tactile feedback, literally the brain is assimilating that as if they were part of the subject's body. And it's representing that in neuronal space, as you saw some evidence for. So we continued looking at that, and you probably may have heard about this illusion that is very famous called the rubber hand illusion that was discovered by a good friend of mine, Jonathan Cohen at Princeton University, and then has been exploited by many, many groups to study this issue of embodiment and self, the representation and sense of self. So if you get any of us here and you put a little platform or a little a piece of wood or plexiglass that occludes the view from one of our arms, and you put a mannequin arm, a rubber hand from any store, and you basically synchronously touch both hands so that the subject only sees the mannequin hand being touched, but the real arm, since it's occluded, even though it's being touched, the guy cannot see it. And after 3 or 5 minutes, 90% of us will say that the real arm is the mannequin arm. You have the feeling that they are touching your arm. And that happens even after this guy stops brushing the real arm and only touches the mannequin arm. This is so real that another group who did a study where they came from behind after they did this illusion with a knife, and they were about to stab the mannequin hand, and the subjects, the blood pressure, the heart rate, the respiratory rate, all exploded because they thought they were being attacked. A piece of them were about to be attacked. So when we saw that, we asked ourselves, do monkeys experience anything like a rubber hand illusion? But since we can actually look inside the brain of these animals, we could go one step further. What happens into the somatosensory cortex when you put in front of these animals 2 virtual arms from the avatar, you occlude the arm like Jonathan did, and you use a little robot to touch a region of the arm at the same time that a robotic device, a virtual device, touches the avatar on the same region, homologous region of the avatar. So you do that for 3 minutes, and you look into the brain. And what happens is very cool. So this is what happens when you are touching the actual body, only the body of the subject. So the S1 cortex, these are cells, this is time. The yellow means lots of spikes. So this brain area, the touch area, is responding because you're touching the actual brain. After 3 to 5 minutes of synchronous touching of the avatar and the real arm, when you stop touching the real arm and only touch the avatar, that's what you see. 30% of the neurons in the touch cortex continue to fire to touch of an avatar that doesn't exist. It's just a visual stimulus. And we know that it's visually driven because the latency of these responses are 16 milliseconds longer than these ones. So we are talking about a piece of somatosensory cortex that for 100 years has been believed to be representing only touch, in 3 minutes being shifted to represent a visual stimulus. 30% is a pretty big number. It may sound small, but it's, for a neurophysiologist, anything of 2% is big. You know? So this is a big effect, which should suggest to us that the animal experienced a rubber hand illusion. He thinks that arm is his, or its arm. Okay? The same is true for the motor cortex. So these visually driven responses appear in the somatosensory and motor cortex. We knew about the opposite. So if you put a blindfold and you go and look at your visual cortex, 20 minutes later, you already start seeing tactile responses emerging on the visual cortex. So if you go blind, for instance, after a few months, a lot of your visual cortex is devoted to touch. And that explains why, you get very good at doing Braille reading. But we didn't know the opposite, that we could drive somatosensory cells with visual stimulation. So, but that can do it. Well, let me just shift gears before I go back to the same topic to show you what is the limit you can do with a brain-machine interface these days. Because as you can see, we rely on 3 main things to get these experiments going. We rely on recording large numbers of neurons. We rely on the ability of the brain to adapt very quickly, to acquire these tools. And we rely on an interface that, believe it or not, is faster than the biological hardware that conducts action potentials in the body. So how far could we take this? So since I needed psychiatric help since the late 90s, I hooked up with a guy much crazier than me called Gordon Sheng, that was at that time in Kyoto, Japan, who is one of the greatest builders of humanoid robots. And he had built this machine with a company here in the United States called Sarcos. It's called CP1, Computational Brain number 1. And this is a perfection of a machinery. And the goal of this machine was to walk autonomous like a human, with one missing detail that the engineers at the end of the process realized they had missed it. They built the joints, they built the body, they built all the beautiful stuff. They missed the brain. How would CB1 walk? And that's where we came. So we said, what about if we lend CB1 a brain by transmitting in real time the brain activity of a monkey walking at Duke University to Kyoto, while visual feedback is fed back from Kyoto to Duke. So this monkey is walking in this virtual reality room and he sees legs, robotic legs, walking as visual feedback while they send the signals to these guys to actually move there. And we actually did that. We realized that the same principles that we saw for upper limb work for lower limb. We start taping these guys. You can make rhesus monkeys walk bipedally. You just need to give support for the chest and the back. And if you have a harness that supports them, they will actually walk on a treadmill very well. So we saw that we could actually predict locomotion patterns very well, actually better with the same number of neurons, better for lower limbs than for upper limbs. And what we did was just send these patterns to Japan. And this is one of the latest experiments there in Kyoto, where CB1 actually walked autonomously, fed by the brain signals coming from a couple of monkeys, in this case is Idoia, at Duke, in real time. 3 beautiful things happened besides this funny walk that the robot did in Japan. First one, the round trip around the world was 20 milliseconds faster than it takes for an action potential to leave Idoia's head and get to her leg. So the stepping of this robot, the robot was stepping on the ground 20 milliseconds ahead of the time of the monkey's foot back at Duke because the feedback was so quick. When we turned off the treadmill at Duke, we still were able to keep Idoia controlling the robot as long as we rewarded her for each correct step of the robot. So if the robot stepped on the curves like he had to do, that was the pathway that we had set, Idoia would get a piece of fruit every step that was correct. She didn't need to move, like Aurora didn't need to move anymore. So we kept this robot walking with a monkey that was just standing still on a treadmill. So that was possible. Finally, this monkey was 5.5 kilos. It was controlling an 155 kilogram of hardware across the globe. So we are scaling time, space, and force with this liberation of the brain from the physical limits of the body. But there was something missing from the whole apparatus that, of course, our monkeys didn't know, but we know, that they had visual feedback. They didn't have tactile feedback. They didn't know exactly when the foot of the robot touches the ground. So I dealt with this thing for many years, trying to, I thought about many different ideas, and finally I came up together with a couple of students with the notion that, well, let's forget about the periphery. Let's deliver the feedback directly into the brain. Let's go to the somatosensory cortex, use the cortex as a computer, create a language, an electrical language, and see if we can make this cortex learn how to interpret tactile feedback coming from the actuator that is under the control of the brain anyway. And to our surprise, it took us 3 years, but it was really fun because we play with creating codes to talk to the brain. It's pretty much like spending 3 years sending messages into the universe and say, is anybody there? Can you answer? And nothing answered, nobody answered. And one day the brain answered and said, okay, I got it. I understand. This is rough, this is smooth, and this is in between. And it was when we came up with these waves of electrical stimulation across the somatosensory cortex that things worked. So this is when we put the avatar that I just mentioned to you in a control loop that was slightly different from what I showed you so far. Because now not only the animal could control this avatar hand to explore the texture of objects, but every time you encounter an object in virtual space, this object, there are 3 objects. They're all visually identical. You could not discriminate which one is the correct one to get a reward. But each one of them had a microtexture, a virtual microtexture, that you could only get by rubbing your virtual finger on the surface. And every time you rub your virtual finger, an electrical pattern proportional to the texture would be delivered directly to the somatosensory cortex, and the monkey had to find 1 out of 3 that if he push it, he'll get a reward. Sounds a little extravagant, but our monkeys are good. So this is, you're going to see in the video clip 3 objects. There's only one that is the correct object. And the monkey, every time the avatar hand gets into this gray zone, the monkey is getting electrical feedback on the somatosensory cortex that is proportional to the texture. There is a high frequency, a low frequency, and a no frequency. And the animal has to identify one object that is moving each trial. And you're going to see legends. The monkey don't see legends. The legends are only for you. So you'll know which one is the correct object in each trial. And what happens is the monkeys actually perform as well as if they use their real fingertips to do this task about 60 days later. It takes 60 days of training. So this is the monkey doing the task. What you're hearing, the background is the brain. And this is the signal that we're delivering to the brain to inform of the texture of the object. So every time the animal recognizes the correct texture, he has to put the hand in the center and press. And then he gets a drop of oranges. So this blip that you hear is a solenoid indicating that they got the juice reward. And they play this for hours a day, and they get close to 95% correct. So they're using their brain to move a virtual hand, and they are touching a virtual object and feeling it through feedback from the virtual fingertips. So now he got it, he doesn't know what he's doing. He's confused. Now he went to the right one and press it. He has 1.5 seconds to choose, otherwise the trial goes away and he loses. Got it again. So if you actually look at these guys doing the task with their own hands versus what I just showed to you, it takes about 4 weeks for them to reach the performance they would do using their own hands. So let me just finish to show you where is the limit right now at all this field. This paper was published last week, just came out in Nature Communications, and it was done in the same lines of what I just talked to you. What is the limit? How much can we dialogue with the cortex, and what is the plasticity level that we may expect? So you probably know that, as I said, somatosensory cortex has been believed to be only related to touch for a long time, since actually neuroscience exists. We asked the question, could we actually impose on top of this touch processing something completely different, something the rats never experienced before? And as you probably know, mammals do not see infrared light. They do not see because there is no receptor in the retina that detects that wavelength, and they don't feel it, the heat of infrared, because different from snakes, they don't have a skin receptor that actually translates the heat, thermal sensation. So what we did was to implant an infrared sensor on top of the head of these animals and get an electrical output of this sensor delivered directly to the somatosensory cortex, particularly to the area that represents the whiskers, the most important tactile organ of rodents. So rodents use facial whiskers like we use our fingertips. They're as sensitive as our fingertips. So we put this guy, it's an adult animal, and we put this guy in 1 hour a day in an infrared universe where sugar water is coded by infrared. So there's 3 ports in this universe, and as the infrared appears, the animal has to basically go to that port to drink sugar water. They love to do that. But they don't see infrared. Basically, we made them feel infrared. So they learn to touch light instead of see light. And that's exactly what happened. After about 45 sessions, these guys start tracking location intensity of infrared. So you're only seeing this because this is with an infrared camera. But the animal goes to the center of the space, it scans the environment, and when the light appears, it tracks it, and it goes there. What you're hearing is the electrical signal that is delivered to the somatosensory cortex as it gets closer to the target. So these guys develop a scanning behavior, that is this head movement that you're seeing here, and they basically start tracking infrared. So when you look inside the brain, what you saw, this is just the performance. They get close to 100% after 65 days. But when you look at the individual neurons of the somatosensory cortex of these rodents, what you find is that not only they respond to the whisker deflections, as they should, because that's what they evolved to do, but now they have infrared receptor fields. They basically develop a multimodal representation. They can play both. So now I know, I said that 2 days ago, the Superman had a cortical neuroprosthetic device to see X-ray. It was the only way to explain it. They didn't put that in the cartoon, but we now, the mystery is solved. Since this thing is coming out next week in Nature, I'm going to show you briefly, if I have 3 minutes, and then I finish, because this is the limit so far. So I showed you that we could impose infrared, that we could basically use these guys to control devices. How far does the concept of the self goes? How much can I amplify the representation of a body? Well, the latest experiment we did, and as I said, it's going to be published next Wednesday, is to connect 2 rats in a brain-to-brain interface and see whether this guy, who we call decoder, can actually base its decisions only on signals that are coming from this guy that is called the encoder, who is actually doing the job. So we put this guy, for instance, we have 2 tasks, but I'm going to describe I'm going to describe 2. First one, this guy has to use the whiskers to judge the diameter of an opening so that it can go through or not. So there's a narrow and a wide diameter, the narrow they cannot go through, the wide they can. So they put the whiskers on the edges, and they do that in 150 milliseconds, easy. And they nose poke. If this is a narrow, they have to go to the left. If it's a wide, they have to go to the right. They nose poke and tell us that. Well, while the animal is doing that, we record the activity of the S1 cortex of this guy, and we send it to the S1 in real time of this second guy that is called the decoder. And the decoder doesn't touch anything, but he has to go and say narrow or wide. And the reason this is interesting is because if this guy commits error, the encoder doesn't get the full reward. So he gets very mad. And the reason we know that he gets very mad is because in the next trial, his reaction time goes down 20%, and his signal to noise in the cortex goes up almost 50%. So this guy gets it in the next trial for sure. So when this guy gets it, this guy goes back to the lazy mode to see if he can get the reward with less effort. But in any event, we established this connection. This is just the second task. The second task is using vision, and it's the same thing. There are two potential visual stimuli that tell either press the left or press the right bar. The encoder gets that. We record the brain activity from motor cortex, and we deliver to the motor cortex of a decoder that doesn't get the instruction. He only gets the pattern of brain activity from the first animal. And he has to answer based on that pattern. So we are using the decoder's brain as a pattern recognition device. And that's what you see in a couple trials here. This is a left trial. So the light is telling the encoder, press the left bar. The guy is seeing the light, presses. Now the signal goes to the decoder. Both lights are on, but he has to go to the left to get the reward. And he's there, he presses the left and gets the reward. To show you that there is no spatial bias, you can see the right trial, where the guy now got some information to press the right. The signal is micro-delivered through a micro-stimulation pattern to this guy, and he's going to go, his reasoning, said, okay, maybe I should go to the right. And they both get the reward. Interesting enough, if you look at the brain of they do about 68 to 70% correct, which is above chance level for both tasks. But if you look at the brain of the decoder after 40 days, you see that the decoder now responds not only to the stimulation of its own whiskers, but there is now a representation of the whiskers of the encoder in the guy's brain. So there are two bodies represented there. The animal's own body and the guy who is giving him the hints where to get the food for nothing. Okay? So it's pretty plastic. Much more than we thought before. So this is all to show you that in addition to studying the brain, these things are coming together, all these lines of work and this multidisciplinary interaction to build a prosthetic device, I hope, that may allow severely paralyzed patients to actually regain mobility. And the approach we are taking after this almost 20 years of work is to actually build a new body. So we are building an exoskeleton, a full body exoskeleton that a paralyzed patient is going to wear. And this exoskeleton is going to ultimately be controlled by the brain. So the movements are going to be as you saw before. This exoskeleton is coated with what we call artificial skin. These are flexible PC boards that have sensors for pressure, temperature, and proprioception. And they are going to provide feedback to the patient's brain, so the brain of the patient will know when the ground is reached, when the knee is bent, or other joints are moving. And basically, we have been working for the last 5 years with this international non-profit consortium of universities in Europe, here, and in Brazil, to actually build this thing. And we just got, you won't believe it, but it's true, we just got permission from the International Soccer Federation, FIFA, who actually runs the World Cup, and a grant from the Brazilian government to actually have the first demo of this exoskeleton on the opening game of the World Cup in Brazil, June 12, 5 o'clock, 2014, 5 o'clock Brazilian time. And the idea is that the opening kick, the kickoff of the World Cup would be a paraplegic young adult, Brazilian young adult, that would walk in the field and kick off the ball. using an exoskeleton controlled by some sort of brain-derived signal. We don't know which one we're going to use yet. So this is the exoskeleton that Gordon, the same lunatic who did CB1, is building. It's a 128 degree of freedom piece of machinery that is really something. This is a prototype that we use in the lab. We're starting to test our monkeys on it, and it's just very simple at this point. These are pneumatic actuators. We are going to use hydraulic actuators in the real patient, and we are just using some signals that we've recorded to actually try to reproduce locomotion patterns in a monkey that is trained to stand up using a harness that supports the body. We are doing a peridural block in this guy in a few weeks, so he's going to be paralyzed for a few hours because the spinal cord is going to be anesthetized, and we'll be able to see if we can get him walking with the exoskeleton controlled by the brain. And this is basically what we hope to happen, hopefully. And if it doesn't work in the World Cup, we have a plan B as the opening ceremony of the Olympics in Rio de Janeiro in 2016. So this is Gordon's rendition, computer graphics rendition of the exoskeleton. Of course, the colors are right. If there's any Argentinian in the audience, forgive me, but and that's what we hope will happen. Believe it or not, after 2 years of this thing in Brazil, people are talking about neuroscience when they talk about the World Cup, which is already enough of an accomplishment. To have kids stop and talk about science in addition to football, it's plenty of reward. So just to finish, then I would like to thank all these kids that over the years have worked in some of these projects that you saw. Michael Lebedev has worked for the last 12 years with the monkeys, together with Joseph O'Doherty, that is now at UCSF as a postdoc. He did the virtual avatar. Miguel Vieira is just publishing the Brain to Brain. Tim Hinson did our interface, our wireless 2000 channel wireless interface. Eric just published the infrared rat study. Laura and Gary work with us since the beginning of the lab. Romulo is in Brazil doing some of the work on the Brain to Brain. Gordon, of course, is our roboticist who is building all these amazing machines. And the guys who helped us at the EPFL in Lausanne to build our avatars, including a human avatar that we're using to train patients down in Brazil now, Hannes Bloor and Suleiman Shakur. Thank you very much.